Welcome to the PubMed Daily Review from May 9th, 2026. Today we scanned 3,046 new open access papers. Here are the top 5 selected for detailed analysis. Trend 1 of 5, Diverse Biomedical and Health Studies. Investigating disease mechanisms, advanced diagnostics, and therapeutic interventions to improve health outcomes across diverse health challenges. This cluster covers 672 papers. Representative work includes functional characterization of the 9q34.13 locus identifies RAPGEF1 as modulating risk for melanoma and nevi via RAS activation, existential distress in advanced cancer. Study protocol of a pragmatic randomized controlled trial of a short-term psychodynamic therapy, or PHYS. Compared to usual psycho-oncological treatment, TU, and cough audio recognition for early detection of respiratory diseases, algorithm development, and validation study. Trend 2 of 5, Sensory motor control and AI. This trend integrates understanding human sensory motor control with developing advanced artificial intelligence techniques. This cluster covers 331 papers. Representative work includes effects of multisensory integration training on postural stability characteristics and fall risk in older adults. Systematic review and meta-analysis. Examining sensory systems that contribute to falls in Parkinson's disease using computerized dynamic posturography, secondary analysis, and FEDI-MONET. Privacy-preserving federated learning with TC and transformer fusion for cross-corpus speech and motion recognition. Trend 3 of 5, Healthcare interventions and system governance. These papers examine diverse healthcare interventions and the governance frameworks crucial for their effective, ethical, and sustainable implementation within health systems. This cluster covers 687 papers. Representative work includes from feminine hygiene to intimate cosmetics. Why language matters for vulvovaginal product exposures and regulation. A sustainable lifestyle intervention among office workers. Cluster randomized pilot and feasibility study, and rethinking post-discharge intervention evaluation. Trend 4 of 5, Clinical interventions and drug studies. These papers explore the efficacy, safety, and practical impact of various clinical interventions and drug therapies. This cluster covers 794 papers. Representative work includes low risk of ocular toxicity following topical ocular administration of epinephrine nasal spray. Implications for unintentional exposure. The Ethics Committee of the Italian Society of Anesthesia, Analgesia, Resuscitation, and Intensive Care (IRTI). Artificial intelligence in end-of-life decision-making processes, ethical reflections, and reply to, is the role of eosinophils in refractory chronic cough truly dismissed? Mepolizumab reduced blood and sputum eosinophils, but not eosinophil-derived neurotoxin. Trend 5 of 5, Environmental engineering and applied science. Papers apply advanced science and engineering to analyze, model, and optimize environmental and diverse real-world systems. This cluster covers 562 papers. Representative work includes performance and mechanism analysis of DMP, 30 modulated waterborne epoxy resin emulsified asphalt, coupled numerical simulation and strategy optimization of land subsidence caused by groundwater extraction in Langfang City, and data-driven fuzzy information granulation for predicting freight volume trends. Up next, our first paper out of 5, Discovery of BBO 11818, a potent and selective non-covalent inhibitor of on and off KRAS with activity against multiple oncogenic mutants by Carlos Stallet and Pedro J. Beltran from BridgeBio Oncology Therapeutics, South San Francisco, California. Let's break down its key contributions. Today, we're diving into a critical area of cancer research. While we have made strides in targeting specific KRAS mutations like G12C, many other aggressive KRAS mutants, such as G12D and G12V, still lack effective targeted therapies. This paper addresses that significant unmet need by exploring the development of a novel pan-KRAS inhibitor. The researchers embarked on a comprehensive drug discovery effort. They utilized various KRAS-driven cell lines and advanced to in vivo KRAS mutant xenograft models to test their novel compound. The core of their design was to identify a molecule that could potently inhibit multiple KRAS mutants by binding to a specific pocket and inducing conformational changes, thereby preventing effector binding. The results were truly exciting. BBO 11818 showed remarkable activity, targeting multiple KRAS mutants including G12D and G12V, in both their active and inactive states. Crucially, it exhibited high selectivity for mutant KRAS over other RAS proteins. In the lab, it potently inhibited MAPK signaling and reduced cancer cell viability, and even more impressively, it led to significant tumor regressions in animal models. Furthermore, combining BBO-11818 with other agents showed enhanced efficacy. This discovery is a major step forward in cancer therapy. BBO-11818 offers a promising new avenue for patients with KRAS mutations beyond G12C, addressing a critical unmet need. While the preclinical data are compelling, it's important to remember that these findings must be validated in human clinical trials. We eagerly await the results from its ongoing Phase 1 trials, which will determine its safety and efficacy, paving the way for potential new treatment strategies for various KRAS mutant cancers. Up next, our second paper out of 5, Beyond Bactericidal, targeting plasmid-mediated antibiotic resistance with natural product-based plasmid curing agents by Jiao Wang and Kun Liang from Heilongjiang University of Chinese Medicine, Harbin, China. Let's break down its key contributions. Antimicrobial resistance, or AMR, is a growing global health crisis, responsible for millions of deaths annually. A major driver of this problem is the rapid spread of resistance genes via plasmids. Traditional antibiotics often worsen this by creating selective pressure. But what if we could disarm bacteria by removing these resistance plasmids, rather than just killing the bacteria? This paper explores a promising new approach, plasmid curing using natural products. This paper conducted a comprehensive review of existing literature, focusing on natural products as novel agents for plasmid curing. It systematically analyzed how these compounds work, comparing their genetic disarmament approach to traditional bactericidal strategies. The primary goal was to elucidate the molecular mechanisms by which these natural products eliminate resistance plasmids and restore antibiotic susceptibility, all within the critical context of the global AMR crisis. The review highlighted the dire scale of the AMR crisis, with 1.27 million deaths directly attributable to it in 2019 and projections of over 39 million direct deaths by 2050. Crucially, it detailed how natural products achieve plasmid eradication by disrupting replication initiation, interfering with partitioning, and blocking conjugation. This systematic analysis revealed a diverse array of natural compounds capable of disarming bacteria without the harsh selective pressures of traditional antibiotics. This review underscores the potential of natural products to revolutionize our fight against antibiotic resistance, offering a path to restore the efficacy of existing antibiotics. However, it also emphasizes the need for robust research methodologies to validate true plasmid curing effects. Looking ahead, the integration of advanced technologies like AI and CRISPR-Cas9 will be vital to translate these promising findings into effective clinical solutions, safeguarding our future against superbugs. Up next, our third paper out of 5, an integrated machine learning framework to understand zoonotic spillover emergence across anthropogenically modified landscapes by Yinxing Zhang and Sun Li from School of Environmental Science and Engineering, 12443 Huazhong University of Science and Technology, Wuhan, 430074, Hubei, PR China. Let's break down its key contributions. Zoonotic diseases, originating from animals, pose a growing threat to global health. Human activities, like land modification, are intensifying our interactions with wildlife and livestock, creating new opportunities for pathogens to jump to humans. But how exactly do these changes drive spillover? And can we predict where and why they occur? This study aimed to answer these critical questions. To tackle this, researchers built a sophisticated integrated modeling framework. They gathered extensive global data, from historical spillover events to detailed human modification indices, climate patterns, and livestock distribution. Using advanced machine learning techniques, including stacking algorithms and structural equation modeling, they aimed not just to predict, but also to explain the complex pathways linking human activity to disease emergence. The study's integrated approach significantly boosted the ability to predict and explain spillover events. A striking finding was that intermediate levels of human pressure, not just extreme modification, actually facilitated zoonotic spillover. They also found that indirect anthropogenic effects, particularly through specific cropping intensities, were strongly linked to disease emergence. Finally, livestock distribution proved to be a reliable indicator for identifying high-risk spillover hotspots. These findings have profound implications for global health. They show that zoonotic spillover risk is widespread, extending beyond traditional tropical hotspots to areas with significant human modification. This research provides crucial insights for developing targeted surveillance strategies, allowing us to focus resources where they're most needed. Ultimately, this work underscores the power of advanced modeling to unravel complex human-environment interactions and better prepare us for future pandemic threats. Up next, our fourth paper out of 5, Serologic Evidence of Highly Pathogenic Avian Influenza H5N1 Virus Infection in a Veterinary Professional Exposed to an Infected Domestic Cat. Los Angeles County, California, December 2024 to January 2025 by Aisling Vaughn and Annabelle D. St. Maurice. Let's break down its key contributions. Highly pathogenic avian influenza A, H5N1, is a serious concern, spreading widely among animals. We've seen spillover into mammals, but a critical question remained. Can this virus transmit directly from domestic cats to humans? This was an undocumented risk, especially given the close bond we share with our feline companions, making it a vital area for investigation. To answer this, researchers investigated 139 individuals in Los Angeles County who had been exposed to 19 H5N1 infected domestic cats. They conducted a serosurvey on 25 of these individuals, looking for antibodies to H5N1. This approach allowed them to detect past infections that might have gone unnoticed, providing crucial insights into potential transmission events. The findings were striking. Out of 139 exposed individuals, 25 agreed to serologic testing. While no one tested positive by RT-PCR, one asymptomatic veterinary professional showed clear serologic evidence of H5N1 infection. This single case, representing 4% of those tested, provides the first serologic documentation of cat-to-human transmission, highlighting the possibility of silent infections. This study is a landmark, offering the first serologic proof of H5N1 transmission from cats to humans, even in an asymptomatic individual. It underscores the urgent need for enhanced infection control in veterinary clinics and for pet owners to be vigilant, especially regarding raw food diets for cats. While the sample size for serology was small, these findings are a wake-up call, guiding future public health strategies and research into this evolving threat. Up next, our fifth paper out of 5, Abi3, S212F, Alzheimer's disease variant alters plaque structure and disrupts microglia by Claire Butler and Kim in Green from Department of Neurobiology and Behavior, University of California, Irvine, California, USA. Let's break down its key contributions. Alzheimer's disease is a devastating condition, and understanding its causes is vital. Microglia, the brain's immune cells, are crucial in AD. We know a gene called MS4A6A, expressed in microglia, is linked to AD risk, but its exact role remained a mystery. This research aimed to uncover how MS4A6A influences AD pathology. To investigate, researchers used a multi-pronged approach. They performed a large meta-GWAS and analyzed human CSF biomarkers. For mechanistic studies, they utilized a specialized mouse model lacking MS4A6D human microglia cell lines and post-mortem AD brain tissue. Advanced techniques like high-resolution microscopy and biochemical assays were employed to examine cellular and molecular changes. The findings were quite impactful. They showed that MS4A6D deficiency significantly reduced amyloid clearance in the brain. Microglia exhibited diminished ability to engulf amyloid plaques, leading to increased plaque burden and more severe synaptic damage. Crucially, this deficiency also exacerbated inflammatory responses in brain cells by disinhibiting NF-kappa B signaling, highlighting a key mechanistic pathway. So what's the big picture? This study reveals MS4A6A as a crucial neuroprotective factor in microglia, directly impacting amyloid clearance and inflammation. The exciting implication is that enhancing MS4A6A levels could offer a powerful new therapeutic strategy for Alzheimer's disease. This opens a promising avenue for drug development, though further human validation and clinical trials are essential to bring these insights to patients. That's all for today's PubMed Daily. If you found this helpful, Please like, leave a comment, and subscribe for daily paper summaries. See you tomorrow.